started out with interests in physics before he figured out that there are actually a lot of really interesting ways to apply physics to answer questions about organisms living in saltwater. So he did his PhD working on bivalve carrying capacity. He got a postdoc working on bivalve carrying capacity, and he continues as a scientist with Canada's Department of Fisheries and Oceans. It's equivalent to our National Marine Fisheries Service, or our NOAA, working on questions of bivalve carrying capacity. And when people come to him and say, how many oysters or how many mussels can I grow in what kind of spacing in a place and still make money on it in a timely way, he has the answer.
Say it in French.
Yeah. Thank you, Jennifer. No pressure now. Before I start, I think I'll take a minute to thank Washington Sea Grant for the invitation. It's really a pleasure to be here and try to contribute to this process. It's really inspiring in some ways as well. And also the organization, Brent, Alex, Jackson, for a warm welcome. Nice activities yesterday. Really nice way to discover the region. So thank you. And what I will be doing in this presentation is giving a fairly generic overview or introductions to carrying capacity and its definition, different definitions, concepts, and trying to show you a few tools that people have been using to assess some of these aspects of carrying capacity for bivalve culture. And I will start. Try to start. Yeah. This is just a very reminder of with what we've been talking about this morning, how bivalves are ecosystem engineers, and so bivalve aquaculture is actually ecosystem engineering. And it's also striking illustration of the filtration of these animals. And filtration, I'll start with that in the next slide, is for this talk is really more of a means to an end. It's how we started thinking about carrying capacity. And because of this obvious effect of particle removal in the water column. So this filtration of bivalves has been studied for a long time, and you can find information about pretty much all cultured species in terms of filtration or clearance rate. And the way it's traditionally done is within laboratory setting, and you can see one of these poor oysters alone in his feeding chamber at the bottom there, trying to feed on what's in the water. And so that's the way it has been done for a long time, trying to follow the diminution in food in this chamber and converting that to the amount of water that animal is able to remove or to filter. So even if it's been studied for a long time, there's still an active field of research. And the recent, I think, work is more towards a qualitative aspect of what type of particles, what size of particles these animals are able to remove from the water, which has a repercussion on the effect they will have on the ecosystem. There's also a bit of work done on trying to be as automatic as we can in measuring these clearance rates. And the reason is to be able to repeat these measurements as much as we can to capture the variability, because it can be fairly high, even for a single oyster, depending on when it's moved. And then the last field that's very active as well is trying to translate these lab measurements into what's actually happening in the field, and finding ways to translate that, or actually measuring these filtration rates in situ in the system directly. Several of my colleagues are actually doing this work. I'm more of trying to integrate this information in tools that are then used to try to figure out how many of these animals we can put in a given place. So we've seen this obvious effect of filter feeders, and it's an easy step to make to introduce the concept of carrying capacity. These animals are feeding on natural particles, natural food resources, and these natural food resources are not unlimited. So that's really the fundamentals of carrying capacity. It's that balance between the consumption of food by these bivalves and the renewal of this food. And typically, we have two mechanisms to renew these food particles. One is the primary production by phytoplankton, the microalgae, and another one is the purely physical mechanism, which is the water circulation, exchange of water between places where you have the bivalves and places where you don't have them. So it's really the kind of fundamental framework that we've been working with for quite some time now. And along the study history of these questions, people have started to put carrying capacity to larger or trying to narrow the questions. And so at some point people I guess derived different categories of definitions of carrying capacity, depending on how you approach the problem. And this is a compilation of the four main categories that are considered, going from the easiest or the simplest to the most complicated. And so just from a physical perspective, it's really the space that you can put farms in a given way. So that space would constitute the physical carrying capacity, and it depends on the gear you're using, things like that, and characteristic of the system. And this is mostly relevant in terms of development of the culture activity, or in marine spatial planning context. So next we move to production carrying capacity, which was probably the way the carrying capacity question was introduced, because growers were the first to realize that bivalves could be limited in their growth by the availability of food. So the formal definition is the stocking density that will maximize the production of the harvest. And then we move into categories that are much fuzzier, let's say, or harder to pinpoint, I guess. And so these two last, as Chris mentioned in his talk this morning, are also fairly closely intricated or linked or giving feedback to one another. So if you want a definition of ecological carrying capacity, you can say it's the stocking density or farm density that will cause irreversible change in the ecosystem. And to try to be objective about just ecological questions. Then the social aspects can be, as Chris said, can't really be separated from the whole questions because humans are part of the ecosystem. So there's always a driven force that comes from social questions or concerns that might limit the development of the culture activity. So I'll be, in the next few slides, I'll be showing a few tools that we've been using, and they're mostly related to these three first categories. And starting with a fairly, I'd say it's in between the physical and production carrying capacity. It's a tool that takes advantage of remote sensing, so satellite images, for example, to provide information about the environmental conditions that are relevant for the growth of the bivalves, so food, temperature. And so usually covering a large spatial scale with these images, which is an advantage for, again, marine spatial planning or development of future activity. And feeding this information of environmental conditions through some sort of physiological response of the cultured species to be able to predict a potential production. And this information can be used for development of waves. I guess this has potential also because satellite imagery is fast evolving, increasing in terms of spatial and temporal coverage that you can get. So the application of this kind of tool might evolve as well, and get into more details of, for example, getting closer to shore, and also maybe looking into interactions between different culture areas, things like that. The next tool I selected is really production focused, and was developed by a Portuguese colleague. And the objective was really to have a tool that can be used by non-modeling experts. And so there's I think they developed a web interface that you can use to input two kinds of information. One kind is about the farm itself, so its dimensions and the stocking density. And the other kind is environmental conditions. And because we're here at the farm scale, we're only considering the current that will produce the exchange of water and the food that's available in the incoming water. And they've linked this model that basically predicts production of the shellfish to a few economical tools to try to move just the biological aspect of growth of the bivalve towards managing a culture activity or farm in terms of benefits or return on investment or this kind of questions that the growers themselves could have. So moving on, if we try to now have a bit of a broader look at the role of the bivalves into the ecosystem, the cultured bivalves into the ecosystem. Probably the simplest tool that starts to touch on ecological carrying capacity is this indicator. It's called the DAME index that you might have heard about, developed by two researchers in the late 90s. And it really comes down to the fundamentals I was mentioning earlier, the comparison between food consumption and food renewal, and doing that at base scale, and using characteristic time scale for a given system. These three characteristic time scale are the ones that are shown here. On one side, the consumption is expressed as a clearance time, so the time it takes for the whole cultured bivalves to filter the water volume of your bay. The second time scale is linked to local primary production, phytoplankton production, so the time it takes to renew the biomass of phytoplankton in the bay. And the third one is the physical aspect of the water renewal, the water exchange between the bay and outside of shore, open ocean. And so by comparing these three time scales, you can show which processes is controlling the availability of food. And so one, I think it'd be easier to show on an example that a colleague of mine developed for mussel aquaculture. And so it's on the y-axis, you have this comparison between the clearance time and the phytoplankton production time. And on the x-axis is the area of the bay that is used for mussel culture. And he developed that relation between these two metrics. And when you increase the number of bivalves in the system, you decrease the time it takes for them to filter the whole water volume, so you get this relation. And people have used different thresholds of this indicator. One obvious value is the value 1, when you get, which would indicate that the bivalves, cultured bivalves, are filtering or using the food resources as fast as they are produced. So it would correspond to a situation where the bivalves are really controlling the whole system because there's theoretically no food left for any other organism. Another value that's been used in the past is number 3, which is hard to find a very hard empirical support for that value, but it's just saying that you leave 2/3 of the food available for other organisms than the cultured bivalves. So I'm going to skip on that. Maybe just saying that this heavily relies on the data that you use to calculate these time scales. So the more you have, the better you characterize your system, the better or more reliable your assessment will be. So moving to a more complex tool that I've been mostly working on in the past few years is taking advantage of my physics background, and building hydrodynamic models of the culture base. So that's a representation of the grid of the model. And that model will calculate basically the water movement, the tides, in response to the water elevation, in response to the tides, the atmospheric forcing, and so on, to predict the water circulation. So an example would be the speed and direction of the currents at any given point in the system and through time. It's a dynamic model. And once this is done, we can couple this model with more of an ecological or ecosystem representation, simplified representation of the real ecosystem. And this would be the minimal structure number of variables that you would need to start looking at ecological carrying capacity for bivalve culture. So probably, well, it's a fairly classical model representing the cycling of nutrients in the system, and we're spending a lot of our attention on the culture bivalve aspect of things, using a specific ecophysiological module for that. It's called the dynamic energy budget. It's being used more and more because you can switch from one species to another just by changing the parameters of the model. So very useful in this context. And so I'll skip through that. Maybe just one interaction that we've been starting to work in the last year, I think. That might have ties with what your questions are here in the region, and maybe with a different perspective. Most of our aquaculture is done in suspension, and people have raised concern about the shading of the culture structures on the eelgrass beds. So we're trying to unveil a couple of studies showing that there's actually a response of the eelgrass productivity under this structure. That's a localized effect, and we're trying to use that modeling framework to be able to balance that localized effect with the filtration effect of the bivalves, increasing the clarity of the water in a much larger area actually than the farm itself, and try to see where the balance actually is. So I'll stop that. The rest is things which we've been working on different components of that modeling structure to be able to have a better, finer representation of the functioning of the system and answer different questions about these interactions. So thank you.